Good morning, students. Today we'll start with a new topic, animal cell culture. In this particular session, I'll give you a brief introduction of animal cell culture and the different types of cell culture. Cell culture is a technique of growing eukaryotic cells, mainly human and animal cells, under controlled in vitro conditions. A cell culture originates from cells of a primary tissue donor and is characterized by cell division and other processes such as differentiation, during which these cells can change into specific types of cells that are capable of functions similar to tissues or organs in the whole organism. The different types of cell cultures are primary cultures, secondary cultures, and cell lines. A primary culture can be of adherent cells or suspension cells, and cell lines can be a finite cell line or a continuous cell line. Primary cell cultures. Primary cell cultures are the cell cultures initiated from normal embryonic or organs, tissues, or malignant tissues. The cells are surgically or enzymatically removed from the organs and placed in a nutrient media under controlled aseptic conditions. These cells proliferate under aseptic controlled environment until they reach a confluence, that is, occupy all the available substrate. Properties of primary cell culture. Primary cell cultures contain a very heterogeneous population of cells and exhibit the same or similar morphology, physiology, and functions of the cell from which they have originated. They have a finite lifespan and a limited expansion. Lineage of cells originating from primary cell cultures are called cell strains. Passage of primary cells leads to generation of cell lines. The many primary cell cultures that are used for research are epithelial cells, fibroblasts, melanocytes, endothelial cells, hemopoietic cells, and muscle cells. Based on the morphology and functional characteristics, the cells can be classified as epithelial-like, lymphoblast-like, and fibrinoblast-like. The epithelial-like cells are attached to the substrate and appear flattened, and the cells are polygonal in shape, as shown in the figure below. The lymphoblast cells are the cells which do not attach themselves to the substrate. They are spherical in shape and remain suspended in the media. The fibroblast cells are attached to the substrate but look elongated and bipolar, as shown in the figure below. The primary cell lines can have adherent cells. As the name suggests, these cells are cells which have contact with each other. They grow as a single layer, a monolayer, on the tissue culture dish. Because of this, cell growth is limited. To passage adherent cells, the cells must be released from the dish either by enzymatic process, chemical processes, or mechanical processes. They are derived from tissues or organs of kidneys, liver, lungs, and brain. They are immobile and embedded in connective tissue. The figure here shows the epithelial adherent cells and the transformed fibroblast cells. The epithelial cells look like a cobblestone appearance, while the fibroblasts grow crisscross and pile up when the high density is reached. Suspension cells are cells which do not adhere to each other or to the tissue cultured plate to which they are grown. They are suspended in liquid as single cells or as free-floating clumps of few cells. Many of the monolayer cell lines which have lost their anchorage become suspension cultures. Example, the HeLa cell lines. Passage of suspension cell line is easier. Only a proportion of cell in the cultures are diluted into larger volumes of medium. The primary cell culture can be obtained from lymphocytes. Advantage of suspension cells over monolayer cells is that their propagation is much faster. They do not need enzymatic or mechanical treatments for passage. The suspension cultures have short lag period and the maintenance of suspension culture is easy. Scale up becomes convenient. A homogeneous suspension of cells can also be obtained. The table here summarizes the differences between adherent and suspension cell cultures. It is possible to grow the primary explants three-dimensionally. In few cell lines, which are anchorage dependent, the three-dimensional growth is initiated by first starting a culture in a stirrer flask for suspension culture. The cells will come in contact with each other and stay together to form large spheroidal aggregates within a couple of weeks. Another way of creating three-dimensional primary cultures are by allowing the cells first to be cultured in a dish which has a non-adhesive surface. This will induce three-dimensional growth. After a few days, these cells are transferred into stirred flask where they form multicellular spheroids and can be kept for several weeks. The three-dimensional spheroid cells are when cultured in normal culture dishes can proliferate again and form monolayer cells. The figure below shows a three-dimensional multi-cell spheroid. Secondary cell cultures. Secondary cell cultures are formed when the primary cultures are subcultured or undergo passage. It refers to transfer of cells from one culture vessel to another culture vessel and are provided with fresh nutrients and growing space for continuous growing. The figure here shows the steps of cell passage. A confluent monolayer growing in a flask is taken and the medium is removed. It is then treated with trypsin so as to disaggregate the cells. On incubation with trypsin, the cells undergo rounding after incubation. The cells are then resuspended in a liquid media and ready for reseeding. These cells are reseeded into a fresh flask and incubated at 37 degrees centigrade. The cells are spread out within few hours in the culture dish and then on top of that the fresh media is added and then incubated at 37 degrees centigrade for 7 days. A primary cell culture after undergoing passages become a cell line or a cell stream. Depending upon the lifespan of the culture, they can be classified into two types, finite cell lines or continuous cell lines. The finite cell lines have a limited culture life, frequently 20 to 80 population doubling. while the continuous cell lines are immortal lines. Properties of continuous cell line. A primary cell can become a continuous cell spontaneously or it can be induced. These cells lose contact inhibition and grow at a very fast rate and high plating efficiency. They show a lot of chromosomal aberrations. The table here shows a comparison between a finite and a continuous cell line. A finite and a continuous cell line may differ in their ploidy level, their lifespan, their mode of growth, and cloning efficiencies. The figure here summarizes as to how the different type of cultures are formed: the primary culture, secondary culture, cell line, and clonal cell line. The different types of cells that can be grown in cultures include connective tissues, skeletal tissues, cardiac and smooth muscles, epithelial tissues, endocrine cells, and tumor cells. Thank you.